This is Immuno Africa, a podcast dedicated to spotlighting African immunology research.
The underlying thing for me at the end of the day is that, HIV has always been an interest of mine. And at the end of the day, I do, even if right now I'm not based in Africa, I do get more pleasure from doing things that involve African scientists that are, that I'm working with people based in Africa, and that I'm doing things that benefit people in Africa, because I feel that.
Welcome to the Immunology in Africa podcast. I am your host, Welt Okete. If it's your first time here, kindly support our work by subscribing to this podcast and turning on notifications to keep up with new episodes. We're currently on Spotify, Apple Podcast, YouTube, and other streaming platforms. Dr. Catherine Kibirige joined me on this episode to talk about a journey to setting up the HIVQuant Project, aimed at transforming how HIV is monitored and managed in resource-limited settings. You can learn more about this project by visiting HIVQuant.com.
Right, so my name is Dr. Catherine Kibirige, as we've already covered. I was born and raised in Kenya to Ugandan parents. My parents fled Uganda during the Idi Amin regime. Were around during Uganda's independence, highly educated. My father was an engineer and my mother trained as a sociologist. I went to a local school, Nairobi Primary School, excelled in the sciences. By the time I got to secondary school, my dad had actually emigrated over to the UK. My parents had split up. But he wanted for my sister and I to join him here in the UK. So my mother put us into a British school. And during that time, the situation with HIV became very bad in Uganda. Uganda was the first place in Africa where HIV was officially acknowledged. And the first cases in Africa were reported in Uganda in a district called Rakai. But once HIV came through, that was very devastating. We lost a lot of people, a lot of politicians died. So it was something that was very prominent. And Ugandans within the, Ugandan community in Kenya were also affected. I lost close relatives. So it was something that really impacted me. And because I had a strong science, was my strongest subjects, I really wanted to be involved in HIV research. So right from, secondary school, O-levels, A-levels, I really was interested in becoming involved in HIV research. So did my GCSEs, O-levels, in a British school, and then came to join my dad and, my stepmother, my other family here in the UK for A-levels. And then did biochemistry at the University of Bath, and then was very tired at the end of it. Ended up being a straight through master's program. They changed the program, so I wasn't expecting to come out with a master's, but we ended up having 2 industrial placements. They augmented the course and managed to graduate with a master's, but was very tired after all of that, so decided to go home to Uganda. My mother had moved back from Kenya to Uganda at the time. So I just decided to go and rest for a while. So I ended up volunteering for the Rakai Health Sciences program. And it turned out that they had actually been studying HIV since it first broke out in Rakai and had set up a large cohort study. Were collaborating with Columbia University, Johns Hopkins University, and the US military. The US military, having lots of troops all around, were particularly interested because it was something that was affecting the troops. So I volunteered, and then they offered me a job. They offered to relocate me, and I worked there for 2.5 years, and then got sponsorship to do my PhD in the US, and managed to get into an HIV lab. I studied HIV-induced cell death, bystander cell death. And then was very lucky to then get a job actually with the US military. Very serendipitous how that worked out as well. But it turned out that when I interviewed, the lab that had sent, so when I worked so how I ended up getting the job with the Ministry of Health in Uganda was because the US military had wanted to set up their own cohort. They had wanted to kind of split away from the collaboration that they had with the Rakai Health Sciences program. And the first scientist that they sent out to actually be based in Uganda and start figuring out a separate lab and a separate cohort for them was there at the time when I volunteered. So my first job offer was actually to work with him and help him set up the lab. And they had wanted to start doing the viral load assays actually in country, because when I volunteered for them, they used to send the samples basically to the US and do most of the work in the US. But there was a big push from the African scientists to do more and more of the work actually in Uganda and to build capacity in Uganda. So he was sent out by the Diagnostics and Countermeasures Division. And he was part of the Technology Assessment Lab. And that was the lab I interviewed with after my PhD. So it just turned out that, you know, they remembered him. They now had their own lab at Makerere University and had established their own things. And we're actually starting to do some work with the Nigerian military. So in the initial job offer I got was actually to do some work on a Nigerian cohort that they were just setting up with the military. But that work was actually delayed. And so I ended up just jumping onto a different project that ended up being the basis of what's now become my main work. So when I worked for the US military, they were doing a vaccine trial, an HIV vaccine trial in Thailand, called the RV144 trial. So it's quite a famous trial. It's actually, so far, it's been the only vaccine trial that's shown substantial efficacy. They had about a 30% efficacy of their vaccine, which they didn't license in the end because they weren't able to really reproduce the results. But so far, it's been the most promising vaccine study to date. But as it turned out, there was a new recombinant strain of HIV in Thailand, and they had commissioned a lab that had published this ultra-sensitive HIV quantification assay that they said could detect down to 3 copies of HIV and could detect multiple different strains of HIV. So they had commissioned this lab to do some research on some of their samples from Thailand. But as it turned out, the assay didn't actually pick up or quantify the strain, the Thai strain. So being based in the countermeasures department and with my boss being part of the technical assessment team, the project ended up on my desk. They were saying, look at the sequences, look at the assay, try and figure out why it's not quantifying these samples, see if you can get it to work or redesign it. So that, so I ended up basically looking at the database of their different HIV strains, looking at the sequences that had been used to produce this assay, and actually ended up redesigning the assay, obviously with the supervision of my bosses. I saw how my previous advisor, Dr. Margley, could use it for some of his samples. So we ended up writing a proposal, and I ended up going back to Johns Hopkins for like a second postdoctoral fellowship where I helped, used the new, I guess, assay that I had developed and developed a more ultra-sensitive format so he could study his archival samples. He had some very rare samples there were just very few cells left. And he wanted to figure out where the HIV was in the patients where, even if they were suppressed with their viral loads, still had inflammation and had poor outcomes. And he wanted to be able to really study what was happening in their different T cell fractions. So I was able to develop an ultrasensitive version of the assay that would work for his very limited samples. And that became my second postdoctoral, I guess, position. I presented the work at a conference, and somebody who I'd initially met in Uganda who had worked for the military, now was working in London. Well, had just finished working for the group that I ended up working with in London, and suggested that I present my work to them because she said, I think they'll be interested in what you're doing. So I ended up presenting and then eventually working for the International AIDS Vaccine Initiative. It's a large consortium. All their field sites are in Africa, but what they call the Human Immunology Lab was in London at Imperial College, and they run all the T-cell based immunological tests for vaccine trials here in London, and not just for HIV or not just for IAVI's vaccines, but any other vaccines. So they've been involved in testing Ebola vaccines and Lassa fever vaccines. But I managed to convince the director to let me finish. So this was a side project that I had carried along with me. Yeah. But I managed to convince the director to let me finish validating the assay. And I convinced her that, it detects all these different strains. And I think it'll improve your vaccine screening assay that you have here. And luckily, she actually had quite a lot of funding and was able to let me complete the experiments to really prove that the assay is very sensitive and works across all the different strains, including the strain in Thailand and some other problem strains that the military have in their repository. So as it turned out, because she'd done a lot of work in Africa and knew the US military HIV team, including their director, she was able to get them to approve me using the original assay I had developed with them. Because when I left the military, they had said that I couldn't use the assay, but they had allowed me to use all the various protocols. But they allowed me to continue that work, and they released all the data, and we were able to finish the validation. And because it worked very well, we were able to disclose it as a new invention. And so when I finished my work with IAVI, that then became my next project. My current project was releasing the assay as a new invention and then figuring out how to commercialize it. And my main focus right now is to develop it as an ambient temperature kit for early infant diagnosis and treatment monitoring. It can work everywhere. It can work internationally, but I'm really I'm focused on improving treatment monitoring and early infant diagnosis or early detection of HIV in Africa, in resource-constrained settings, in remote settings, where currently there's a huge delay in people being diagnosed, and then there's a delay in their treatment monitoring. So that's basically the story of how I've ended up coming to set up the HIVQuant Project, is what it's called. And that's my current HIV-related work that I'm doing.
I know you've covered most of the projects you've worked on and how you got into them, what they led to, but I would just like you to briefly highlight some of the, maybe just a kind of high-level overview of some of the outcomes of those projects. For instance, your PhD project, and then the postdoc that you were involved in. So what was like the, in a simple way, what was like the question you're trying to address? What did you find out? And then what was the impact that some of those answers held?
Okay. So with my PhD, I was looking at bystander cell death. So I was looking at the effects of, not the direct effects of the virus when it infects the cells, but the effects, because most of the variants that are released from infected cells are actually defective. And then you also have bits of the surface protein that end up in your circulation, but they will bump against immune cells and kind of activate them and just cause this inflammation. So most of the cell death occurs. So, HIV is known for causing a lot of inflammation and for particularly depleting CD4 T cells. But most of those CD4 cells actually aren't infected. It's just that they're interacting with on the surface, or even if some of the pseudovariants can get into the cell, but they're not well formed enough to actually have a very productive infection. But even that, just that interaction with the surface proteins or with the pseudo-virions activates cells and sends them down a cell death pathway. So most of the cell death is actually occurring in bystander cells. And I wanted to look at these mechanisms more. My hypothesis was that the bystander cell death actually also occurs in the naive cells. So your naive cells, if you know, I'm sure you know, I mean, if it's immunologically based podcast, that hopefully your viewers are aware that T cells, naive T cells tend to be, so you have your stem cells, you have your progenitors. The first, I guess, population of T cells are your naive cells. And then once they're stimulated, they develop into the various different subsets of T cells that have specific functions within your immune system. The naive T cells are the ones that can regenerate the most, divide the most, and form all these different T cell subsets. But if you are able to, if there's a virus or something that's able to affect your naive cells, then that's going to have a very profound effect on your immune system. And at that time, the dogma was that HIV did not infect naive T cells, and that bystander effects didn't, I mean, there was not much proof that much was happening with naive cells. But what I did show, even if later there was some evidence to show that, okay, some naive cells can be infected, they do express CD4 and CD8, it's something that's still, I guess, kind of debated, although maybe not so much in the field. But I was showing that actually you don't even need to infect the naive T cells. There are bystander effects that will affect the function, cause partial activation, and may also affect their ability to regenerate. So the bystander mechanisms are actually affecting the naive cells, and that's what's causing the really detrimental issues on your CD4 T cells as a whole, because you couldn't account for what happens in your CD4 subset just by direct infection. So I ended up not having enough data, like a control experiments, to prove this without a doubt. So in the end, what I published was a hypothesis paper. I had data to show that, just from looking at my samples, and just from, I had done experiments where I exposed, how did I set them up in the end? I exposed the cells, I was using supernatants. I was using infected cells within the lab, and then harvesting CD4s and CD8 cells from different donors, and just showing that I was getting cell death in the naive T cell compartment, that was due to bystander effects. And so that was published as a medical hypothesis. But one thing I really got from my PhD, which it showed me that I wanted to do more translational research. I wanted to do more research that had more of a direct effect on the patients rather than something that was more theoretical. So I think when I moved to the technology assessment lab and was working more on improving diagnostics and involved more in vaccine trials, that to me was more satisfying than doing more kind of mechanistic work. So that's what came out of my PhD. I have the hypothesis paper has been cited quite a few times, and a lot of the theories I had within the hypothesis paper have now been proved by other scientists. So there's now much more evidence to show that naive cells can be infected. They will express or they will upregulate CD4. Like, there are mechanisms by which they can be infected, but even without the infection, just the bystander mechanisms of all these virions and all these products is having a detrimental effect generally, but also affecting the naive cells and their ability, the CD4 naive cells and their ability to regenerate. And this is what's causing detrimental effects on the immune system.
So I would like us to move next to the HIVQuant project that you've been on. And you've highlighted it earlier on when you were talking about your journey, but I want us to kind of shine the spotlight more on it now. So let's talk about what the HIVQuant project is about, and tell us what it's about, and then what inspired you to take up that kind of project to help improve HIV diagnosis or HIV quantification.
Right. So like I mentioned, it started off as a side project. The development of the assay that's underlying, I guess, the project started off as a side project when I worked for the US military's HIV program. And I kind of carried it with me through 2 more postdoctoral positions. And then it actually ended up becoming the one really good publication that I got out of my third postdoc, and then ended up becoming the focus of my work for the last year and a half. So, like I said, when I worked for the US military, it wasn't really the project I was employed to work on, but serendipitously, I ended up being involved in investigating a quantification assay that hadn't worked very well, and figuring out how to improve it, and then actually trying different cycling protocols and reagents, and actually figuring out how, well, it turned out to be very sensitive on its own, but we did various manipulations with the cycling and investigated various reagents that actually really made it a very sensitive assay. So I was able to go on to do some ultra-sensitive quantification for part of my second postdoc. And was actually able to quantify HIV in naive T cells. So when I went back for my second postdoc, I was actually able to go back to these archival samples from my PhD lab and show that actually HIV could be quantified in the naive T cells. And for people who were immunosuppressed, so were viral load undetectable, but were still were still experiencing inflammation and were not having as good outcomes as other people, there was actually a significantly more higher amount of HIV in their naive T cells. So I was actually able to circle back and actually come back and prove some of my hypothesis, and actually got to present that work at a conference. But the really exciting thing out of all this work was this new assay that was very sensitive, that works just as well as current gold standard viral load assays. But I guess with the HIVQuant project, my focus is more to produce an ambient temperature kit and figure out a system for the use of the assay in either secondary or primary treatment centers that currently don't have the capacity to do the viral load testing, so that early infant diagnosis via DNA quantification and treatment monitoring can be done closer to the point of care and can become more efficient. So I guess I'm moving from more translational research to even more like public health and public health implementation in resource-constrained settings. And that's kind of where I'm heading with my career. My motivation basically is because there's a huge gap. I think once with academia, a lot of what we do once you get to a certain point is trying to figure out where the gaps in the field are and figuring out where you can find a niche and where you want to, I guess, focus your energy, focus your career. So when I finished my postdoc, it became quite evident when I worked for IAVI that I'm not that interested in vaccine research. I found like the diagnostics and being able to use this assay to address this gap in the field and being able to finally go back to Africa and actually do something impactful and something positive with all my education was more appealing to me. So that's what birthed the HIVQuant project, and that's what's motivating me to continue the work. We were granted we were able to file and get a provisional patent and then a global patent treaty on the assay. So the big challenge for me right now is to get more funding to really get the kit to work. I've done a feasibility study in Malawi. I was able to get some funding to do that, which was great. But I tested 2 sets of reagents, and one of them didn't keep very well at Malawian room temperature. And these reagents are still evolving. So I've had companies send me their different formulations as they're trying them. So this is something that's with these reagents, it's just I guess a modification on the enzymes that's just coming out now. The companies are able to get the reagents to work at room temperature. But obviously room temperature in the UK is quite different from room temperature in Africa. So they're still improving the reagents. And there's a lot of new platforms that are coming out that are more portable. When I went up to Malawi, we went out with a small magnetic induction cycler that really doesn't draw much power and can work on a solar panel. And that's what we tested. But there are now other platforms that are smaller and simpler. We actually have a collaborative agreement with a company that has a very, a card, it's, I can't say too much because a lot of it is proprietary, but it's something that could potentially be done by a nurse with a finger prick. So we're looking at novel and interesting platforms that could potentially work in resource-constrained settings where there's no reliable electricity and there's no refrigeration. And that's the focus of the HIVQuant project.
Yeah, thank you. Thank you so much. So at what stage is the project now? You already mentioned that you've done feasibility studies. So what stage are you on in that project?
So at the moment, it's a bit of a side project again, because I run out of my initial funding. I managed to get on an accelerator program, so a business accelerator program where you're funded to do market research and try and figure out, where, if there's a need and where in the market you can position yourself and where you can enter into the market.
Yeah.
And the overwhelming advice I got was that it's not at a point where I can set up a company. I need to develop it further and just find a unique platform that fulfills the function that I want to fulfill for the assay. At the moment, I'm actually working in a different position. I'm working as a research manager, but in the background, I'm putting together proposals and trying to find teams who can help me further the work. So another recommendation I got was that I need to move into a group that has more experience with developing and commercializing diagnostics and doing field trials in Africa. So when I did my feasibility study in Malawi, was quite interesting. I ended up having to present to the hospital team there, and it took a long time to get the study approved, and a lot of it was because I hadn't really designed it very well. So I'm having to, I guess, kind of go back and kind of do more study epidemiology. I'm moving more into public health and epidemiology and public health implementation. So it kind of challenged me and made me realize, actually, I need to go back and do a bit more study and just kind of consolidate my knowledge in that area. But I was advised that I need to be in a group or have a principal investigator or a sponsor who has that expertise and can help me move the project in the direction that it needs to go. So I was invited to speak at a new diagnostic symposium where I got to meet various researchers who do that type of work. And I've been reaching out to them, telling them about the HIVQuant project and offering to present my work from Malawi. And I'm basically just trying to find the right group of people to work with to help me further the work and put out proposals to get more funding. I'm at the stage now where I try to become independent as a scientist. And to do that, you need to, I guess, get grant funding in your name or within a group that's doing similar work. So that's what I'm trying to do at the moment.
Yeah, thank you. I understand it's a lot. So trying to piece together the aspect of business, trying to make money from it, and of course, the fulfilling that goal, the public health angle of ensuring that people are able to access these new diagnostic, and then it's able to change the current landscape of how HIV is diagnosed and quantified in low resource settings. So thank you for sharing the challenges. I also noticed you've already highlighted your next steps, so what you hope to look at accomplishing next on the project. So next, I want us to bring the conversation a bit down to Africa. I know you mentioned that the feasibility study you conducted was done in Malawi, but I'm just wondering if you had already, or maybe you plan to have collaborations with institutions or individuals in Africa, because if your this innovation, you are targeting low resource limited settings like we have in Africa, I understand that it's important to work with even those who are based in Africa to ensure, understand the challenges, understand the risks or the experiences of individuals who have HIV in the context of the continent to actualize your goals. So what's the current plan for you? Do you already have individuals you're collaborating with or institutions you are collaborating with on the continent, or do you plan to explore such collaborations?
Well, I'm well plugged into, I guess, the HIV network in Africa, having worked there. I still have ties with old colleagues at Makerere University. If you look at our website, HIVQuant.com, you'll see that I have got other members of my team. At the moment, most of my team members are based in Uganda. So I still have very strong links with Makerere University. Dr. Sabiti, who's another member of our team, he's based here in the UK, but he's done a lot of TB work, including having a TB assay licensed by a medical charity. He does a lot of work in South Africa, and he actually has an organization set up in Uganda that does clinical trials. So some of the proposals that we're putting together is to do some work with Dr. Sabiti. There's a lot of interest from various colleagues in Africa. I've actually been approached by a WHO reference lab in Uganda, in Kampala, that would like to test the assay as soon as we have a version that's ready for validation. So, then apart from the Malawi Ministry of Health, I do have colleagues in West Africa and people who've offered to introduce me to people even in Nigeria. But it's just like, one step at a time. I do want to have a viable prototype first, and then take it from there. But I am plugged into the HIV network in Africa. I was talking to people within the WHO. So apart from HIV, they want to know if I can do the similar thing for other neglected tropical diseases. I've had people reach out to me about EBV, for example. So I think with the platform, at the moment, it's an open platform. So once we get the system working for HIV, then there's nothing to stop us from introducing other diseases. HIV is highly challenging, it's highly variable. So if we're able to get it to work for HIV, then adding other assays should be relatively simple. We just need to get the initial assay and the initial system to work. So yeah, I am looking to collaborate. I am interested in obviously field testing, but interested in novel platforms that would work for resource constrained settings, and also funding opportunities. People who want to collaborate, who potentially would be interested in funding the work, and who have ideas or contacts for companies or groups that have a platform that would work in resource constrained settings that the assay could be used with.
Yeah, sure. Our listeners would get that, and I believe and hope you get all the support you need very, very soon. Yeah, thank you so much. So I believe you've had rich or robust experience with mentorship, because from your background, you work with several individuals, even work with individuals, the same individuals in different settings. So, so I would like you to just talk about how mentorship has contributed to your journey as a scientist, and perhaps how you made efforts to give back to society, leveraging that platform of mentorship.
Right, so mentorship is very important. I actually was interviewed for International Women's Day and wrote a blog about why women scientists need mentorship, because there's a lot of discouragement, and at the moment within STEM, subjects of science, technology, engineering, mathematics, women are very underrepresented. At least here in the UK, ethnic minorities are underrepresented. The further up you go in, institutions like Imperial College. So, I've found as my career has progressed, I'll go to conferences, and many times I'll be the only, well, there'll be very few females. Many times I'm the only ethnic minority, and certainly many times I'm one of the, and unless it's more of the global international conferences, but when you get to at least the more technical ones, or even with the business kind of accelerator programs, I found that many times I'm the only ethnic minority, and many times I'm one of very few women. Like with the last accelerator program I did, it was only 2 female-led ventures, and I was the only, African-led venture. But having mentors who've encouraged me despite all of this has been really key, because along the way I've always had people saying, "No, not possible, not possible." There's been a lot of blocks. It's really been a lot of hurdles to climb, and just having people who can encourage me, people who've gone ahead, doesn't have to be a woman, doesn't have to be a ethnic minority. Many of my mentors have been, one of my mentors actually was an Italian doctor who works for GSK, who actually just picked up an interest and really encouraged me, helped me improve my CV, and gave me a lot of tips and pointers. So mentorship can come from anywhere. But I think, especially as a female minority scientist, I've just found that the mentorship that I've received has really been crucial and important, because there's been so much discouragement, and there's no way I would have gotten to where I got to now if it wasn't for people who really just took the interest and just said, no, you need to keep going, that you actually have something important here, and who just really took the time to say, look, this is how you present yourself. This is what you need to do. This is how you need to navigate this system. This is how you need to navigate this particular situation. And I still have mentors even now, because I'm still coming up against various challenges. At the moment, I'm getting a lot of mentorship in grant writing, translation, getting a lot of help in, figuring out my next move and which group, how to position myself within my current institution, because there are other people doing similar work. So how do you find your niche? How do you remain competitive? How do you navigate yourself in these types of environments? It's very important to have somebody mentoring you.
So I'd like to ask about your experience as a scientist who has worked in Africa, and also you've worked in different settings in the diaspora. You've worked in the UK, you've worked in the US. So to you, what are some of the gaps or challenges? Of course, I believe some of the challenges are quite obvious in the African setting, but from your own perspective.
I had always wanted to go back to Africa. It's funny, because I was always one of those people, even when I came here to do my A-levels, my focus had always been to get what I'm getting from the West and go back home. And, I got teased a lot about it, and I was a bit of an odd one out in my family. But then when I went home, and some people said, it's best you go home because then you'll appreciate the West more. And when I went home, there was a lot of things I appreciated about it, and it really did open the door for me to enter HIV research. I think, getting to work on an HIV project and advance as much as I did I wouldn't have been able to do that in the West just because of the barriers. I think going back home, having had my biochemistry degree from the UK, and originally I went as a volunteer, so it was more about giving and service. That was something that really impressed the people back home, and they really did put me forward for the sponsorship scheme that I eventually got onto that led me to the US. So that was really good. But one thing I found that was when I was then ready to go back, there was a lot of antagonism because, as I'm sure you're aware, back home, there's a lot of really, really qualified people competing for very few well-paid jobs. So there was a lot of physicians who had been working on the project for a long time, who then went on to, do the PhDs, do lab-related work. So I was competing against, physicians who had a lab background to become a lab manager, a lab director in a pool of lots of very qualified people. So that was very frustrating. I think, one of the big frustrations going back home is there's a lot of really, really qualified people going for very few jobs. So, when you go for interviews, you're competing against, physicians who have Masters of Public Health and, have a MD, PhD, all these letters after their names. And it's, it's very disheartening. So it's been very hard for me to go back for those reasons. And then there's a lot of corruption as well. There's a lot of nepotism. So that's frustrating. And so then it becomes a question of, okay, how do I collaborate? If I'm based here and I'm still interested in doing work back home, then I need to find ways of collaborating or gear my research towards Africa. And then that way I'm not competing or taking away from the people there, but, having impact and helping, I guess, elevate the work of people that are working there. So at least currently with the proposals that I'm hoping, I'm collaborating with people back home. We're hoping to have students on the projects and that will help them advance. But even that in some ways is frustrating because, like at least here within Imperial, there are other scientists already doing that. So trying to find my niche, and trying to compete for funding, while being based here, and then work more with scientists in Africa is also proving to be a bit of a challenge. So, but I'm getting there. It's been a long road, but I'm hoping that I can do more work in Africa in a way that's mutually beneficial for all of us, and that does, I guess, bringing the best of both worlds, trying to, leveraging the advantages I have of being based here, access to maybe funding opportunities, business training, industry collaborations that people back home may not necessarily have, and having a perspective that maybe people back home won't have, and coming in with a unique angle and managing to work with scientists on the ground and move things forward. But it's been interesting and it's been challenging. I think my heart is in Africa. So as much as, I've tried to settle here, and as much as there's been a lot of challenges with trying to go back home, I think, what the underlying thing for me at the end of the day is that, HIV has always been an interest of mine. And at the end of the day, I do, even if right now I'm not based in Africa, I do get more pleasure from doing things that involve African scientists that are, that I'm working with people based in Africa, and that I'm doing things that benefit people in Africa, because I feel that, I've had these advantages, I've had this education, it would be nice to be able to pass some of those things back to the continent. So it's still very much a passion of mine. But they are lots of challenges. I mean, you know, you're based in Nigeria. I'm sure you understand the frustrations of the corruption. You understand the frustrations of the high unemployment. Lots of very, very qualified people. I mean, I'm always very impressed with the scientists that I meet back home. I've been very impressed with the African students I've interacted with, I've met here. The African student I had so far was one of the best students I've had. So we have very qualified and able people back home, but it's just the challenge of, I guess, the poverty and corruption and the things that we have to deal with on the continent that make it difficult for people who are based here who want to come back and give back. Yeah. It would be easier to just settle here and do work here, but I don't think I would get the type of satisfaction I get from being able to do things that are impactful and improve the lives of people in Africa.
We've had so far a very robust conversation, and I think you've had a very inspiring journey and also a very inspiring story from how you got into HIV research and where you've taken yourself, or where you know that singular project you started out with in the field of HIV has brought you to now trying to develop a very sensitive assay or platform for quantifying HIV, and you know that will be not be limited by location or the setting or technological advancements that can be used almost anywhere. So I think that's really an inspiring story and inspiring work you're doing. So to wrap up the conversation, I would like you to share a closing remark or your final words on the episode. So you're allowed to share anything at all. So maybe a technique that you've learned, something that has helped you, a word of motivation, or just anything at all that you'd just like to share with the audience, who could be students, who could be immunologists, who could be just curious listeners.
Well, I would just encourage people to think out of the box and to draw inspiration from their surroundings. You know, I'm very inspired every time I come back to Africa, because people have a can-do attitude, and people are having to make ends meet and make things work, I guess, out of nothing, you know. And I mean, there is a lot of corruption, etc. But I'm always really inspired by how entrepreneurial and innovative and creative people are, just to survive. And I think that's where people should draw their inspiration from. If you are, I mean, coming and getting your education abroad is great, but it's not necessarily the answer. I think people really need to look at what's going on in their local environment and see what they can do based on what's around them. You know, just agriculture. You know, I mean, even here in the UK, people are realizing the need for, you know, natural products, organic products. Africa is so full of resources, and we have very people and very healthy people, and that's something that's actually as the years go on, you know, the population in the West is becoming more and more unhealthy despite all the advancements and all the technology here. We're relying more on our organic, like more on more natural foods, and we're doing better health-wise. So that should really tell you something about, you know, the richness in Africa. We have a richness of resources. A lot of our land is still very unspoiled, and we really need to look at that and find inspiration in that. And even where we do have challenges, I think a lot of the solutions are going to come from what we have locally. And, rather than looking too much to the West, we really need to look more to what we have on the continent, what we have, what resources we have in the continent, and what we have within ourselves to provide solutions for ourselves, And I think that's where we'll win.
Thank you for listening to this episode on Immuno Africa. If you enjoyed it, please feel free to share it with your network. You will learn first about future episodes and get other immunology related updates by following Immuno Africa _ or the Immunology in Africa podcast on social media. See you on the next episode. Bye.